Welcome to another exciting video. This is DR.BYSON Muntari bringing you nothing but the best in medical courses. Today we're looking at epithelial 2, exocrine glands. This is histology practical number 4. Without wasting much time, let's look at what we are doing today. Alright, so on the introduction, what we need to know is that epithelial cells make up glands. So being either exocrine glands, which possess a duct, or endocrine glands, which are ductless. Glandular epithelia is also known as glandular tissue, and this refers to a type of epithelial tissue involved in the production and in the release of different secretory products. Okay, so now glandular epithelia is arranged into structures that are known as glands, which can either be a single cell or a group of cells specialized in producing and releasing a specific substance. For example, sweat, saliva, breast milk, digestive enzymes, and hormones, among so many other substances that glands are able to produce. Now let's look at exocrine glands. Exocrine glands produce and secrete a large variety of fluids that are important for digestion, lubrication, lactation, and thermoregulation. Examples of the most common exocrine glands include sweat glands that are found in the skin, salivary glands that are found in your face, in your mouth, lacrimal glands, and mammary glands. Okay, now this schematic diagram is showing us how an exocrine gland is shaped. So this portion here is called the secretory portion. It's made up of acini cells, secretory portion. So this secretes what your glands produce. And this is the conducting portion. It has ductal cells. From the name itself, it conducts what has been secreted by the secretory portion to the surface epithelium. Now, the most important question that one would ask is that how are glands formed? Now, glandular epithelia is formed during fetal life by the development of epithelial cells into connective tissue that underline the epithelia. So initially, all glands are connected to the surface epithelia by a channel called a duct. However, later on, each gland will differentiate into either exocrine or endocrine depending to the presence or the absence of glands, respectively. What this means is that exocrine glands have a duct and endocrine glands do not have a duct. Now let's look at gland classification. How do you classify glands? So glands are classified based on number one, the type of their secretory portion. Okay. The shape of their secretory portion. Secondly, either whether their duct is branched or is not branched. So this is a schematic diagram showing us the different types of glands in our bodies. So this one here is a simple gland. Why? Because the portion here is not branched. The secretory portion here is tubular. So this is a simple tubular gland. Here it is branched. So you can see the conducting portion here is branched. So this is a branched tubular gland. This gland here is coiled. So it is known as a coiled tubular gland. This one here, the secretory portion is oval shaped. So when it is oval shaped, we say it is acini or alveoli. So this is a branched alveoli gland. As you can see, this gland here is highly branched. So it's a compound tubular gland because its secretory portions here have got a tubular shape. So this is a compound tubular gland. And here the secretory portions have got an acini shape. So this is a compound alveoli gland. And the compound alveoli glands mostly found in air sacs, which are found in the lungs. Now let's get into the lab. What are the objectives for this lab? Number one, to classify glands on the basis of their morphology and types of secretions. Secondly, to describe the structures of different types of exocrine glands. And thirdly, to enumerate or explain the factors that stimulate or inhibit secretions by various glands. So the first thing that we're looking at is simple columnar or simple tubular glands that are found in the ileum. So what we're going to look at in this slide of the ileum is that we're going to look at adjacent villi. So between adjacent villi, they are what we call straight tubular glands. Straight tubular glands which are continuous with crypts of Leboucan. So between two villi, there are crypts of Leboucan, and these crypts of Leboucan continue and form glands. And these glands at the base have got red granules which are called Paneth cells. So now it will be very important for you to write the function of these Paneth cells. So what you are going to do in the simple tubular glands is that you're going to draw the simple tubular glands at high power and at low power. Okay, so this diagram here is showing us the low power representation of simple tubular glands. Okay, so this, if you remember from our previous lesson on epithelia 1, this is what we call a prica seculare. So a prica seculare is going to form villi. So this here is a villi, this is a villi, this is a villi. Now between two villi, there's a villi here and the villi. So this is a villi here and the villi here. Between these two villi, there's a crypt. The crypt is continuous as a gland. So this is a gland here, this is a gland, this is a gland, this is a gland, this is a gland, this is a gland. So you can't really appreciate them well here because this is low power. Now let's try to look at them at high power. So draw this at low power and then move to high power. This is high power. Draw it at high power. So at high power, this is the gland here. So you'll notice that these glands are lined by simple columnar epithelia. Okay. So this is a gland here. This simple columnar epithelia here, simple columnar epithelia. So the simple columnar epithelia line these simple tubular glands that are found in the ileum. Okay. So this diagram here, we cannot appreciate the Paneth cells, but let me show you another diagram that has got a beautiful representation of the Paneth cells. All right. So these are our glands here with simple columnar epithelia, and at the base of the gland, these red granules, these are the Paneth cells. Paneth cells, Paneth cells, Paneth cells. These Paneth cells are the ones that secrete your intestinal secretions. So write the functions of the Paneth cells. This diagram gives us a very vivid picture of what we were talking about. So this is a villi. Okay. So this is a villi, and then this is a villi. Between these two villi, you find this crypt here. This crypt is what we call a crypt of Leboucan. So these crypts of Leboucan will continue and form glands here. These are glands. So this is a gland. In histological diagrams, it appears like this. So this is what you are drawing at high power. So draw this, label these as simple columnar cells, draw this, label these as simple columnar cells. This is your Paneth cell, this is your Paneth cell. So for simple columnar glands, draw at high power 1 and at low power 2. Okay. Now let's move on and let's go to the uterus. Now for the uterus, you look for tubular glands extending from the lumen of the organ lined with simple columnar epithelia. And some of these glands may be branched. Your task is to draw at low power and high power. So we're going to look at the uterus at two stages. One will be your secreting uterus and the other one will be at a non-secreting stage. Okay, so this is at low power, non-secreting uterus. So this is the uterus. This is the uterus here, and this is the lumen of the uterus. So as you can see, this is the gland, and this is branched. So this is a gland, this is a gland, this is a gland, this is a gland. These are glands here. You can't see the epithelia that lines these glands because this is at low power. At high power, this is how it appears. So this is the uterus at a non-secreting stage. This is the high power. So this is a gland here. This is a gland. This is a gland. This is the more clear gland. So this gland here is lined by simple columnar epithelia. So make sure you indicate that this is simple columnar epithelia that is lining the gland. So draw this gland here. Draw this gland. Show this connective tissue and draw this gland here. That's all. Now let's go to a lactating or a secretory nucleus, uterus. So you can see these are glands. The glands in a secretory uterus are wider and they are tortuous, meaning they are irregular and they are wide. The reason why they are wide is because they are big. Okay, so they are secreting here. Secreting, that's why they have got material inside. They're secreting glycoproteins and everything that is needed for the baby to grow. So here, this is at low power. You draw secretory nucleus, the glands here. So you just pick 1, 2, 3 glands and just show the connective tissue that is surrounding. At high power, this is how it appears. You see now this you can see. So this is the gland here. And you can see the cells that are lining the glands. This is simple columnar cells. They are very dilated, and inside here there are glycoproteins, complexes that are being secreted by the gland. And this is another gland. So draw the uterus in secretory phase at high power, low power, and draw a non-secretory uterus at high power and low power. Now let's go to sweat glands. So sweat glands are found in the scalp. So we're looking at the scalp, slide 35. So what we want to do in this slide is that we want to identify sweat glands, which are coiled tubular glands, which secrete by merocrine methods. Merocrine methods is where your glands are secreting by exocytosis. There are 3 modes of secretion. The glands can either secrete by merocrine, apocrine, and horocrine. So you need to know these 3 types of secretions by glands, and they will surely show up in your histology test. So the task is to sketch at low power and to sketch also at high power. Now let's look at these glands. Okay, so your sweat glands are coiled. So these are your sweat glands. So this is your sweat gland here. You can see they are coiled here, here, here. These are your sweat glands. That's your sweat glands at low power. At high power, they will appear like this. So this is a sweat gland here. This is a sweat gland here. This is a sweat gland here. So that is at high power. This is a sweat gland there. So that's what you are looking at. So the sweat glands are lined by either simple or pseudostratified columnar epithelium. So make sure when you're drawing these glands, you show the type of epithelia that is here. This looks like it is pseudo-stratified columnar epithelium. All right, this is another diagram. So this is a sweat gland. It is coiled and it has a secretory portion here. So it's coiled. The conducting system is coiled and the secretory portion is here. So in fact, this is a very good diagram to draw your sweat glands. So draw this, include the secretory portion here. This is also a sweat gland here. It's coiled like this, and the secretory portion is here. All right, now in the same slide, we'll also identify sebaceous glands. So sebaceous glands, how you identify them is that they are usually attached to the hair follicles into which they empty. So you observe the epithelial capsule surrounding the gland with changing in size of cells. So you have to pay attention to the frothy nature of the cytoplasm resulting from dissolved lipid droplets. So these are circular glands which secrete by holocrine secretion. Holocrine secretion is where an entire cell bursts or is damaged so that it can secrete its substances. Alright, so the sebaceous cell is this one here. Sebaceous gland is this. So this is a medium power sebaceous gland. Okay, so these are the cells that are inside of the sebaceous gland. So you draw here and you label, and then this is at high power sebaceous gland. So this is your conducting portion here, and then this is the secretory portion. So these cells will be bursting so that they can be secreting their substances. That is how sebaceous glands produce their secretions, by holocrine secretion. Now let's move on to our mammary glands. What type of glands are in the breast? So we'll look at the breast both in the lactating phase, when a woman has a child and they are breastfeeding, and in a non-lactating phase. So now, what you should note is that glands are secreting and therefore alveoli are dilated. When the glands are secreting, the alveoli are dilated with homogeneous pink-stained material. So inside, there are lipid droplets there. So the alveoli are lined with simple cuboidal epithelia, whose lumen is uneven because glands secrete by apocrine. So mammary glands secrete by apocrine. These are the things that will show up in your MCQ, so you're supposed to know. So in this slide, you identify ducts, observe what their epithelia is similar. You observe that their epithelia is similar to that of the lining, the ducts of sweat glands. So you draw at low power and high power. So now the gland that I'm about to show you here is a mammary gland that is secreting. Okay, so this is in your mammary gland, so it is made up of, this is your lobule, and this is your lobule. What you find between two lobules, these are what we call interlobular ducts. Interlobular ducts. And inside of the lobule, you've got intralobular ducts. So you show all of these. These are intralobular ducts. These are interlobular ducts. So this is a lactating mammary gland, because you see the lumen here are dilated and they've got colloid material inside. So this is low power, you can't really appreciate. If we look at it at high power, this is the high power, lactating. So when you look at the lumen of the glands, which are lined by simple cuboidal epithelia, they've got a substance inside here. This substance is what we call colloid. It is mucus. So you know your mammary glands produce mucus. So they will be inside of the lumens of the glands, mucus. What we find between the glands are intralobular ducts. So draw this at high power and low power. A non-lactating mammary gland is identified by a lot of dense connective tissue. See, there's a lot of dense connective tissue. So these are the ducts. Okay, these are the ducts that lead to the glands. So these are the ducts, and between the ducts there is a lot of dense connective tissue. Therefore, you know that this mammary gland is non-lactating. This is non-lactating. Now, if we get a portion of this and we project it in high power, this is how the gland is. So the gland is lined by stratified columnar epithelia or simple cuboidal epithelia. Okay, so this is this looks like stratified cuboidal epithelia, and it has a lumen here. You see that in the lumen there is nothing, meaning it's not producing anything. There's no mucus here, so it's non-lactating. And the lumens here are empty, non-lactating. Lumen empty, non-lactating. So for the breast, make sure you draw a lactating breast at high power and low power, and a non-lactating breast at low power and high power, using these images that you're able to see. Okay, now let's look at the serous and mucous acini. So these glands are mixed glands. These glands that are found in the sublingual gland. Glands that are found in the sublingual gland. So you identify, compare, and contrast the two types of acini labeled at high power. So this secreted by merocrine secretion. So this is your sublingual gland at low power. So low power, this is a lobule, this is a lobule. Between two lobules you have your interlobular duct, and inside of your lobule you've got your intralobular duct. Now when you look at your lobule, there are these portions that are appearing whitish. These are what we call mucous acini. And then there are these parts that are appearing purple. These are what we call serous acini. So this is a mixed gland because it has both serous and mucous acini. This is at low power. At high power, it will appear like this. So you see, these ones that are appearing pale here, these are mucous acini. These that are appearing purple, like this, these are serous acini. The ones that appear pale are mucous acini. The ones that appear purple are serous acini. This here is an interlobular duct. So it's between two lobules. So how do I know this is an interlobular duct? Because surrounding it here is connective tissue. Okay. So there's connective tissue. There's connective tissue between lobules. So this is a lobule here. This is a lobule here. And this is another lobule. These two lobules are separated by this. Okay, so this is connective tissue, and in the connective tissue you find ducts which are interlobular ducts. All right, so this is just high power showing you the serous acini. So 6 here is the mucous acini, pale stained, and then 3 here is the serous acini. Okay, 4 is the serous demilume. Okay. All right. So this is just a schematic diagram that is showing us the serous and the mucous acini with serous demilume. So the ones that stain pale here, these are the mucous acini. The one that stain purple, these are the serous acini. And then they've got myoepithelial cells. Myoepithelial cells are cells that contract to squeeze the gland so that it can release its secretions. Okay, so that was the last one. To further study this chapter, go to Janquilla chapter 4 and you have a better understanding of this. So if there's any exercise for this, it was surely given in your lab, or it will be sent to the class rep and you will answer. Otherwise, this is a very simple lab. Always make sure to refer to the video on where you are not clear. This is Dr. Bison EM, bringing you academic data in excellent form. Let's medicine together the Unza way. I'll see you in the next video.